Good morning, ladies and gentlemen. So it is my great pleasure to introduce Professor Wallace McKeehan from Texas A&M University. And actually, there are many postdocs from Hiroshima University worked in his lab. So far, I know the 7 or 8 associates now working in graduate school of biomedical science has been in Wallace lab. So, and I'm happy to have his lecture in this workshop. So I briefly introduce his CV. He received his undergraduate degree in chemistry at the University of Florida in 1965. And then he got a PhD in biochemistry at the University of Texas, Austin in 1970. And he was a research scientist at the Basel Institute of Immunology in Basel, Switzerland, from 1971 to 1974. And research associate in the Department of Molecular, Cellular, and Developmental Biology at the University of Colorado from 1974 to 78 at, I think, Dick Ham's lab. And then he joined to the W. Alton John Cell Science Center from 1979 to 1993 in Lake Placid, New York, where he was a senior scientist, deputy director, and co-founder of Upstate Biotechnology Incorporation. And while there, he held a adjunct faculty appointment in Department of Biochemistry at the University of Vermont, and Department of Chemistry at Clarkson University, and Cell Biology Department in Xiamen University in China. And he is currently the J.S. Dunn Professor and Director of the Center of Cancer and Stem Cell Biology at the Institute of Biosciences and Technology, Texas A&M Health Science Center, Houston, and professor in the Department of Biochemistry and Biophysics at Texas A&M University. And he was named the Texas A&M Regent Professor in 2003 and Texas A&M Distinguished Professor in 2008. So I hope we could continue the Old Boys Network who has been in the Wallace lab in the future. So Wallace, please.
I will, unfortunately, for most of this audience today, I apologize for it. This may be a rapid-fire presentation and I don't expect you to catch every point of it, particularly with my Texas accent of English, which is sometimes harder to understand than a non-native English speaker's accent. But I'd like to go through and tell you some of the things that we have done, and we've been doing a little bit of history, not to bore you too much with it and bring you up to date of it's sort of our view of the FGF signaling system, as well as some of the most recent things we're doing that have opened up in the FGF receptor system. And I understand that this will be recorded or so, or you'll be able to see these slides later. So don't try to capture everything I'm going to show you here, data and all that. I hope you'll get the overall viewpoint of it. So the FGF signal transduction system, as I said in the opening title, is involved now in two major areas of homeostasis in organisms: cellular homeostasis and metabolic homeostasis. This is the historic area on which FGFs have been most widely studied. And by cellular homeostasis, we mean the balance of cell types and function within tissues primarily in embryonic development, where everything is changing continuously, but it also is important for compensatory homeostasis, growth when needed, but also limited growth and establishment of function in adult tissues, which I'll talk about mostly later, the liver, which we like a lot. And Dr. Matsubaru will be talking about some of the applications in the prostate, which we've worked with historically in our laboratory. FGFs were discovered in pursuit of endocrine factors in cell culture, although as a cellular homeostasis factors, they were not classical hormones or endocrine factors. They ended up to be tissue growth factors, or maintaining cellular homeostasis within tissues. Ironically, in about 2000, it opened up with some of our studies, and then several other labs picked it up immediately. It's involved in endocrinology and metabolic homeostasis. And ironically, this is the balance of specialized cell functions in organisms and specialized organs. And this seems to be a classical organ-to-organ endocrine communication system. And ironically, this was discovered in pursuit of growth factors in vivo. We discovered the endocrine activities of FGFs. FGF signaling, whatever you work in, you'll run across it eventually. It's ubiquitous, as we said. It's a large family, and it's driven by several members and cofactors. That I'll tell you a little bit about later. That it's multifunctional depending on where you find it. It impacts most cell biological process, and therefore its dysfunction has wide-ranging consequence in context of the individual tissue where the family may be at play. The FGF heparan sulfate tyrosine kinase signaling complex is not agreed upon what it looks like as a complex, unfortunately, and I'll tell you a little bit about that later. This is our view, our model of it. It is a big family. It has 18 receptor-mediated isotypes and some homologs that don't interact with receptor. It's 4 tyrosine kinases that are transmembrane here, the kinase domains inside the cell here, and an ectodomain that looks like this, and it does exist as an oligomer, as I'll tell you a little bit more detail in a moment. It is driven, the cellular homeostasis aspects, to date, heparan sulfate is an integral player, and it's very diverse in terms of oligosaccharide motifs there. And this is a model you'll see over and over in this talk. Here's sort of heparan sulfate, our view of how it enmeshes the complex of the dimeric FGF transmembrane tyrosine kinase here. Now, heparan sulfate plays 2 general roles in FGF signaling, and a lot of people forget heparan sulfate. It's difficult to work with, it's sugar, it's glycobiology, and they take it for granted. But it's a major component and driver of FGF signaling, and it cannot be ignored. And we have given a great bit of attention to that and asking questions of what is its role. It plays 2 general roles. It's a tissue matrix control of stability or reservoir and access of FGFs to FGF receptor, as shown here, out in the matrix or so like this. And it also is an integral subunit of the FGF receptor complex itself. It interacts with the receptor, and it plays 2 roles there, we believe. It contributes to the overall specificity for FGFs as FGF comes into the receptor, and it also exerts conformational control of FGF oligomer and the final signal transduction by the receptor. So it has these multiple roles, and they're very difficult to dissect, and it's very important to try to dissect these different roles because they're all at play in a given system that you might be studying in cell culture, and particularly in vivo. Now, one problematic aspect of the FGF signaling system is we don't know what the receptor looks like on a structural basis. We're mostly biologists here, but structural biologists that have done X-ray crystallography and a lot of other assays, there are 2 irreconcilable models of the FGF receptor. They both can appear in crystals by X-ray crystallography from the same components. So we don't know which one's correct, or if they're both correct, or if they're half correct. One's wrong, one's right. And there's quite a bit of controversy about what the receptor looks like. One model shows a symmetric type molecule here of the FGF receptor ectodomain, and one is completely different, as you can see in this model here. Here's the two models that are not reconcilable. Half of each one kind of fits with the other, but they can't be put together. They can't both be right. Here's the one model that's shown here, and here's the other model. They're totally in different orientation. Heparin sulfate is tying the two together. Here's two FGFs, whereas this one's symmetric, like two FGFs here, and heparin sulfate is coming across as a part of the receptor complex here. Now, a third model was proposed by us to unify these models from a long time ago. It's what we call the conformational model of a pre-existent dimer that is enmeshed in heparin sulfate, and it is held inactive by heparin sulfate, and FGF comes in and conformationally makes a change in the outside domain that is transmitted through the intracellular domain that actually activates the receptor itself in this type of model. So these are the three types of model that I'll be talking about. We don't know which one is exactly correct, but our view, our biased view somewhat, people would say, is the conformational model. It's ready to fire. It's sitting there. Heparin sulfate plays a cofactor role in restricting its activity, and FGF then comes in and changes that conformation and overcomes that activity. And this is just the model of how we think it works. The enzyme-substrate complex relationship is not correct. It's not close enough on the inside, although the ectodomain is already dimerized, ready to fire. And heparin sulfate restricts this orientation, so these are far enough apart so they don't transactivate. Now, the FGF receptor, like many receptors, transphosphorylates itself. That's why you need a dimer. That's why you need two to tango, so to speak, to activate and have an activated receptor. So the FGF receptor is tightly internally repressed by a repressor domain that covers up the kinase active site that has to be phosphorylated by the adjacent neighboring kinase to de-repress and allow substrates to come in and interact with the receptor and be phosphorylated like this cartoon over here. This model was developed by primarily the influence of Dr. Kahn, who's back here in Japan with Xeria Pharmaceuticals, one of our earliest Japanese associates that drove these models and the role of heparin sulfate in the early days. And these two papers is what gave us this idea of the conformational model. He found that heparin sulfate cooperated to control assembly, but it also had a negative effect in absence of ligand. It made the receptor ligand dependent. And then another Japanese associate, Umatsu, did an interesting experiment. He did mutations on the inside domain in here that affected the characteristics of the ectodomain when in a membrane. So that meant that somehow conformationally you do a change in here, it can be transmitted outside to the ectodomain. Well, that's not the way the receptor normally works, but we did argue that, well, if the ectodomain can sense a change artificially in the intracellular domain, then the intracellular domain must be able to sense a change in the ectodomain, a conformational change, and that's another support of this model that we have used. Now, from our laboratories come some of the most extensive experiments, again driven by Dr. Mikio Kahn when he was in our laboratory for many years, and together with several others, that we do believe that beyond just charge density, the heparin sulfate molecule is very specific when it interacts with the FGF receptor complex. And the early experiments found that there was a requirement for best activity for a oligosaccharide that had anticoagulant activity. And that's the most popular or well-known characterized activity of heparin sulfate, as most of you know. That's what heparin does in the clinic. And it's the anticoagulant motif is characterized by the distribution of sulfates across an oligosaccharide rather than just a lot of sulfates. So this was picked up when Dr. Kahn left our laboratory by a very bright glycobiologist or biochemist, Yongdu Lo, who's still with us in the laboratory. And he published a series of very difficult structural papers to argue that there is structural specificity in the type of oligosaccharide that will not only react with a given FGF, but also have activity for signaling of the FGF receptor complex. And as you can see here, this was based on the idea of looking at the face, the heparin binding interface of the different FGFs here. There's 18 of them, and not all are shown here. You can see how this face, here's the most popular, FGF1 and 2, there's a heparin oligosaccharide, how it's different in respect to charge density and also topography. So this argued that there must be a different relationship in each FGF potentially between an oligosaccharide and the FGF binding. And indeed, what he did is to take one of the FGFs and to bioaffinity purify with a very nice, properly folded, highly active FGF like FGF7. And to make a long story short, he broke up heparin oligosaccharides and used the FGF as an affinity reagent itself, and then eluted at different levels of salt, and then analyzed by mass spectrometry and enzyme degradation what the characteristics of those and bioactivity of those oligosaccharides were. And to our surprise, with FGF7, he extracted a very highly purified, with highest affinity to the FGF that had was a mixture of 7 and 8 sulfates. Now, a fully sulfated octamer of heparin is 12 sulfates. So this was an undersulfated oligosaccharide that had highest affinity for FGF based on salt dilution. So we argued that this cannot be just charge density, because this must be some sort of specific interaction depending on where these sulfates are in the molecule. And to make a long story short here, he found that this oligosaccharide was very specific for FGF7, but is activity with FGFR2/3b, but it causes the FGFR2/3b receptor complex to reject FGF1. It will only work with FGF7 and not FGF1 in this case. If you use this highly purified octamer here, this is mass spectrometry of which we use to analyze these, and as you can see, it's much more heterogeneous here with a number of sulfates. We get highly fully sulfated material coming down at lower salt off of this FGF7 than we do at higher salt with less sulfates. That is very specific. So he went on to characterize this a little bit more, and this is his deduced structure. This is not proven. It's very difficult to prove even an oligosaccharide structure now by sequence technology. But he did show by various assays that it has 2 unique groups in it. It has a 3O sulfate that's very rare. It's in the anticoagulant motif. And it has a rare unsulfated amino group as well, an N unsubstituted glucosamine, which is rare also. And so it has those 2 groups in that one octamer, which says there must be structural specificity. So in summary, for the role of the cofactor heparin sulfate, we believe, as we've told you a little bit here, that hidden in all this extensive diversity of heparin sulfate in a matrix or in biology is in the high charge density, hidden by all that, is oligosaccharides with specificity. They have cell and tissue type specificity, and it is a key cofactor in control of FGF signaling. It integrates FGF signaling with the extracellular matrix, and therefore the matrix controls FGF signaling. And heparin sulfate, as we say, in number 5 here. Now, to turn to another area that in the diversity of the cellular homeostasis roles of FGF signaling, we worked for many years with a model of in which usually in the normal case is a paracrine type signaling. It's a cell-to-cell communication system in the local environment, the cellular homeostasis roles of FGF. And here's just a classical example of where one receptor is partitioned in the epithelium and the factor comes from another compartment, the stroma. And that acts over here on a specific receptor in the epithelium, And then most recently, we've found that the epithelium will crosstalk back within the FGF receptor family with another pair in which the factor is only in the epithelium and the receptor for it is only in the stroma. So you have this two-way directional communication system. So I won't spend much time in this area here because Dr. Matsubara picked this up in our laboratory about some of the implications of this system in tumor biology and tumor suppression, and he'll tell you a lot more about that, which I don't have time to tell you about anyway. But to summarize this area, it turned out that the resident epithelial cell FGFR2/3b promotes tissue homeostasis, part of cellular homeostasis, and its loss promotes dysfunction, including tumorigenicity. However, when another homolog isotype of the FGF receptor system appears in the epithelium, it's never there normally. It's activated in stroma and damage in the wound response. It drives the epithelial cells as part of the mesenchymal transition, we call it EMT, it's become popular to call that, and drives malignancy. So whereas FGFR2b promotes tissue homeostasis and actually dampens growth and properties associated with tumorigenicity, FGFR1, if it appears in these same cells, goes the opposite direction. And this was substantiated by a long series of papers of names, some of which are recognizable here. Dr. Matsubara was involved with this in 1997 and 98, and is continuing it somewhat, which you'll talk about today. So one thing that has been really powerful for us, the early ideas here with FGS were the tissue cultural growth factors in cell culture, is to bring those questions into the mouse as a model of human disease and ask the questions, does all this work in vivo? It's fine in cell culture, stimulation of growth, inhibition of growth. And indeed, without going into great detail, Dr. Fen Wang is with us still today, became an expert in mouse genetics technology. And in general, the ideas I've been telling you about so far, at least of which Dr. Matsubara will talk about, R2, epithelium versus R1, has been totally substantiated in mouse models. And so we believe it's not just a cell culture artifact or interesting phenomena of this one growth factor inhibits cell growth, one drives cell growth. When it appears in the epithelium, the mouse models are clearly, and other labs are substantiating this over and over. Now, given that, I want to turn to one of our favorite organs that we worked with from the time that Dr. Kahn came into our lab. He liked liver, and we were working with liver biology. And since we were in the FGF field, we began to ask questions about what's the role of FGF in liver? Is it a growth factor? Does it drive liver regeneration, which is a dramatic regenerative organ if you damage it, resect it with partial hepatectomy, or damage it? FGF as a growth factor has got to be a major growth factor in liver. Well, it turns out after 10,